Thank you, David. I'd like to thank at the outset David, the BPS, and the BHS for this opportunity to present. The topic is difficult. I can't answer the question. So what I'll do is I'll give you a brief perspective on drug treatment of hypertension and focus on two specific examples using genomics and metabolomics to discover novel pathways. As you know, blood pressure has been around for a long time. People knew about blood pressure right from 2600 BC. Even at that time, the Yellow Emperor of China, the books showed a relation between salt and blood pressure. As our understanding of circulation improved, so also did our methods of measuring blood pressure. So with the Rivarochi cuff, we've been easily able to measure blood pressure quickly and in large populations. Along with this, efforts have been made to try and reduce blood pressure. As Gordon mentioned, people, when you see high blood pressure, you'd like to reduce it. And what this slide shows is all the drugs that we use commonly for blood pressure has been discovered and in common use over the last many years. And we don't have any new drugs coming out in the market. So there is a need to discover new pathways. Just to go back a few steps, in history, it was initially thought that high blood pressure was important. This basically came from the recognition when people observed high blood pressure was associated with constricted arterioles, and they felt that you needed the high blood pressure to maintain perfusion through these constricted arterioles, and hence came this concept of essential hypertension. And there was controversy about whether to reduce blood pressure or not. The multifactorial basis of high blood pressure, or the multifactorial basis of blood pressure, came about quite elegantly described by Pickering, who noticed a normal distribution of blood pressure in the population, and he felt that there was no demarcation or a dividing line between people who have high blood pressure as a disease and people who do not. And this implied that the risk of blood pressure was a continuum across the range of blood pressure. In the early 70s, Guyton tried to describe this multifactorial basis of blood pressure by the circuit diagram, wherein he tried to integrate all the known mechanisms at that time. And even now, all these areas are still important targets for treatment. So the kidney, circulation, brain, and endocrine hormones. So all this happened in the early part of the century, and the critical role of the kidney in blood pressure came about with Guyton's pressure natriuresis hypothesis, wherein he felt that, or he hypothesized that the blood pressure, everybody had a set point or an equilibrium point for blood pressure. If your blood pressure increased, then you get something called pressure natriuresis, you get increased sodium excretion, and your blood pressure returns to normal. If you have a shift to the left of the equilibrium point, you have hypertension, and if you have an increased slope of the curve, then that's salt-sensitive hypertension. You don't have an increased slope, it's non-salt sensitive. The shift to the right indicates high blood pressure. Of course, we know most of the pathways. I don't need to describe or enumerate the pathways associated with blood pressure regulation, but all these pathways do have an impact on your pressure natriuresis curve. So angiotensin 2, aldosterone, endothelins, and the sympathetic system tend to push the pressure natriuresis curve to the right, and dopamine, potassium, ANP tend to push it to the left. And if you look at a common treatment for blood pressure, ACE, ARB, beta blockers, diuretics, they tend to block the pathways on the right and thus inhibit the rightward push of the pressure natriuresis curve, whereas you increase potassium or dopamine or give dopamine agonists, you tend to push the pressure natriuresis curve to the left. So whatever be the pathway we use, there seems to be a link to the kidneys. Of course, we all follow a reductionist paradigm. We all want simple explanations. We want to have a single gene, a drug acting on a single gene that can affect your blood pressure. And most of our data on single gene studies come from quite successful experiments in monogenic syndromes for hypertension. And this is the seminal work from Lifton, wherein he discovered a lot of these monogenic syndromes for hypertension. And what was clear, all these syndromes tend to be clustered around the kidneys and the adrenal gland, or the final common pathway appeared to be the kidney. though originating from the adrenal gland. And most of these syndromes do have specific treatment. For example, Liddle syndrome is a syndrome due to persistence of the epithelial sodium channel in the collecting duct, and you have a treatment for that. You block the epithelial sodium channel with amiloride and you reduce blood pressure. We also know of complex pathways like glucocorticoid remediable aldosteronism, which seem to be driven by ACTH, so giving glucocorticoids paradoxically is a treatment for this condition because it reduces the ACTH drive. So using a reductionist approach, you could identify genes, you could identify receptors, pathways, or enzymes that affect blood pressure regulation, and we have drugs targeting those specific molecules leading to a reduction in blood pressure. And genetic studies have expanded our understanding of the molecular pathways or the signaling pathways that regulate these, and we have we know most of our existing drugs. We now have a detailed understanding of the underpinning pathways, but we are just blocking the final common pathway. So for thiazides, we are blocking NCC. We do know syndromes like Gitelman syndrome or Gordon syndrome, which are due to mutations in the NCC channel. The treatment is the same. ENaC, epithelial sodium channel in the collecting duct, tend to be is important for the aldosterone signaling pathway. The treatment is either blocking the epithelial sodium channel or using aldosterone antagonists. In the thick ascending limb of the loop of Henle, you have important sodium channel, sodium transporters, and mutations in them is known to cause a Bartter syndrome. And an effective blocker of the NKCC2 in the thick ascending limb is the commonly used furosemide or any loop diuretic. The problem is we need to identify new drugs. We know that we are all aware of the problems of getting blood pressure to target. We need more drugs. We need to identify probably novel pathways for blood pressure control. So one way to identify novel pathways is through an agnostic or hypothesis-generating approaches exemplified by genome-wide association studies or any other high-throughput studies. And Nilesh explained the basis of genome-wide association studies and the complex challenges facing once we discover signals. This is the work led by Mark Caulfield and Patricia Monroe on blood pressure genetics, and they've discovered almost 35 to 40 significant hits for blood pressure. 2 points about these Manhattan plots. One is the genes which are indicated here these do not mean that the genes have been conclusively linked to the SNPs. The signals are an alphanumeric code for a SNP associated with blood pressure. We just find the nearest gene, or through some bioinformatic method, we try to assume that the nearest gene or something which is an LD with a SNP is the associated gene, but it's not truly so. We need a lot of biological experiments to try and link the SNP to the gene and gene function. So that takes a lot of work. We have come a long way from the page model of hypertension. If you incorporate all the genetic studies and genome-wide association studies, the page model can now be represented as such. What we want is more clinical translation. Let's go back to history again. If you look at all the monogenic syndromes, these arrows describe the time of description of the monogenic syndrome and the time of gene mapping. And what you notice is none of the monogenic syndromes have actually led to discovery of new drugs. All the drug discovery for hypertension happened independent of genomics. So what we want is, this is the era of genomics. So what we hope is GWAS will hopefully lead to new drugs which are not captured by the pathways we already know. If you look at the pharmaceutical industry's development of new drugs, they are targeting multiple segments of known regulatory and counter-regulatory pathways. And these may lead to incremental benefit to blood pressure response, but these are not going to lead to us targeting new pathways. So the hope is for GWAS and hypothesis-generating studies to discover novel druggable targets which would not be discovered otherwise. And as Nilesh commented, some of these have been discovered in coronary artery disease, but are there any for blood pressure? I'm going to describe a GWAS of hypertension, which we did a few years ago. This was a GWAS of hypertension. So we looked at blood pressure extremes. We selected the top 2% of the blood pressure distribution and the bottom 9% of the blood pressure distribution as cases and controls. This is the Manhattan plot. We found just one signal. We spent about £1 million and got one signal in chromosome 16, which didn't attain genome-wide significance. So we we replicated in multiple studies. We increased the sample size to 39,000, and this showed quite consistent results across all the populations, attaining a genome-wide significant p-value of 10⁻⁻13. So that gave us some comfort that we did have a significant result, but we were not sure whether it was a true signal. We looked at the regional plot, and this is what happens. You get a good signal, you find a number of genes. That's a gene expressed in the testes, that's the gene expressed in the gut, and that's the gene expressed in the kidneys. We assume we're dealing with blood pressure, so this could be an important gene, but we need to have confirmatory evidence. Luckily for us, that gene, uromodulin gene, is only expressed in the thick ascending limb of the loop of Henle, where 25% of the filtered sodium is reabsorbed. So that was a good sign. We measured the uromodulin excretion in the urine in two independent populations, a hypertensive population and a normal general population cohort, and we found a significant association between the genotype and the uromodulin excretion, indicating that our SNP was linked to the gene, and hence that gene potentially could be related to hypertension. We also did a study with Dutch collaborators. They had this study of a salt intervention study in young males, young normotensive males. And what we found if the individuals were on a low salt diet, you had the relationship between the genotype and uromodulin. But once they were put on a high salt diet, the relationship disappeared, indicating a salt or gene environment interaction. So the best way to prove causality is to knock the gene out. So we took a mouse model with the uromodulin gene knocked out. We didn't do anything. This mouse model has been available for many decades. There is a monogenic syndrome of uromodulin associated with kidney stones. So the urologists have been studying this model for many years. They never measured blood pressure. So we got the mouse model and just measured blood pressure. This recovery period is because we implant a telemetry probe for blood pressure measurement, continuous blood pressure measurement. So we measure blood pressure, and without any intervention, we see the knockout mice had a lower blood pressure compared to the wild type mice, and that fitted in with our GWAS results. We then fed these mice salt, and we did the pressure natriuresis curve. In the wild type mice, we found a linear relationship between blood pressure and sodium intake. In the knockout mice, there was a shift to the left of the pressure natriuresis curve, indicating that these mice are salt resistant and they do not show an increase in blood pressure with salt, indicating a possible salt mechanism in blood pressure regulation through this gene. So what does this gene do? This gene is only expressed in the thick ascending limb of the loop of Henle. It's trafficked by the Golgi apparatus to the cell surface. It is anchored on the cell surface through a GPI anchor. So it requires a specific protease to cleave so it actually lines the cell membrane on the luminal wall. You need a protease to cleave the protein out, and the moment it's released into the lumen, it polymerizes into a gel which moves down the nephron. So that's our study in knockout mice. After we published our paper, there was a group in Italy who have been doing a lot of work on uromodulin. They did some nice experiments on a knock-in model wherein they overexpressed uromodulin. So the opposite of what we had done. And they showed after overexpressing uromodulin, the increased uromodulin, overexpressed uromodulin mice had higher blood pressures. They looked at sodium transport with overexpressing uromodulin, and they found increased expression of NKCC2 in the thick ascending limb in the overexpressed mice. They looked at uromodulin and NKCC2 activity, and this is an interesting study wherein they transfected human embryonic kidney cells with uromodulin, and they showed an increase in NKCC2 activity. They also transfected with soluble uromodulin. So soluble uromodulin do not get anchored on the cell surface. And they showed there was a decrease in NKCC2 activity, indicating that you needed the uromodulin on the cell surface, anchored on the cell surface, to affect NKCC2 activity. So NKCC2, as I mentioned, is a target for loop diuretics. They had data so this group from Italy, they had data in normal or treatment-naive hypertensive patients wherein they had given a sodium load and given furosemide. And when they looked at the blood pressure response to furosemide based on genotype, they found the uromodulin increasing genotype had a better response to better diastolic response compared to those with the uromodulin decreasing genotype. So you have uromodulin increasing genotype, you have increase in NKCC2 activity, and hence you're a better responder to loop diuretic. So what we need to prove this. So we are in collaboration with David and Tom. We've designed this trial. We're just waiting for funding if it comes through. What we decided we would look at in a randomized, or what we thought we'll do is we'll give toracamide, which is a long-acting loop diuretic, and look for genotype differences in blood pressure response. If that is the case, then we could repurpose loop diuretics for uncontrolled hypertensives in individuals who have the uromodulin increasing genotype. We don't normally use loop diuretics in hypertension, but if we can show efficacy of this by genotype, then this would target about 67% of the population who would have the uromodulin increasing genotype. So that's ongoing, awaiting funding. One interesting study so this story has been always interesting in terms of gene-environment interaction. So I mentioned the genotype uromodulin association with salt intake. Now, when we finished our GWAS and got this SNP in the uromodulin gene, we were quite surprised. At the same time, we were quite shocked, because the previous year, the Charge Consortium had produced their GWAS on CKD. And they found a series of SNPs associated with kidney function. And one of the top signals was a SNP in the uromodulin gene. And this SNP was very close to our SNP and in 100% linkage disequilibrium. So there was a problem here. We thought maybe our results were confounded because we had just discovered a SNP for good kidney function, which led to a better blood pressure. So we did correct for various things and EGFR in our study, and we did show an independent effect, but still we couldn't get away from the fact that we could have our blood pressure SNP was actually a CKD SNP. So this is the Framingham data. They show increase, the genotype with decreased uromodulin is associated with increased GFR and a reduced rate of CKD. And we did show the same result in our population. We did show that our SNP was associated with a slightly higher GFR. So we did this, we measured kidney function in our knockout mouse model. So this is the wild type mice, we did nothing, and this is the knockout mice. We measured creatinine clearance, and paradoxically, we showed the opposite effect. In the UMOD knockout mice, GFR, creatinine clearance, was actually lower than the wild type mice. So this was opposite in direction to the Charge Consortium meta-analysis for blood pressure. When you feed these mice salt, you get no change in creatinine clearance in the wild type mice, but you get a significantly higher creatinine clearance in the knockout mice. So this would indicate that the GFR association with the SNP with UMOD is associated with, or is related to salt intake. And it's very likely that the human GWAS studies relating our SNP to kidney function is probably confounded by the high baseline salt intake in the population. So we wanted to look at this more clearly in the humans. So this is again the Greco study. These are normal males, normotensive males who are given high salt and low salt. And when we looked at the low salt arm and the high salt arm, we saw the opposite effect. So if you're on a low salt diet, you get the same relationship between genotype and uromodulin excretion, whereas if you're on a high salt diet, you get the opposite effect. So that proves that there's confounding, and this is a blood pressure gene and not a CKD gene. So we now have a pathway. As I mentioned, there are monogenic forms of uromodulin, which leads to uromodulin-associated kidney disease. These are mostly in exons 3 and 4. They lead to defective UMOD trafficking, leads to ER stress and damage, ESRD and death. These do not cause hypertension primarily. It's hypertension related to kidney failure. These GWAS SNPs are present in the promoter region, and they lead to mainly UMOD expression level changes and pressure, sodium homeostasis, problems with sodium homeostasis. The effect on GFR probably could be explained by tubular-glomerular feedback. So where does our SNP lie in this ICBP Manhattan plot? It lies somewhere here. So this is the GWAS for blood pressure as a quantitative trait. And our SNP is nominally significant. It's about 10 power minus 3, so it'll be here. It doesn't really cross the threshold for genomic significance. So this also indicates that when you do these large genomic association studies, you do get the top-line results, but there are also possible causal variants hidden underneath, which are probably hidden because of the dilution in the effect size or significance because of all the confounding factors. So that's the genomic pathway. I'm just going to quickly go through another hypothesis generating experiment using metabolomics. Now, genomics, we do a complete screen of all the genetic variation across the genome. Metabolomics, we look at small molecules. So this was in collaboration with TwinsUK. It's a large cohort of twins who already had metabolomics done using the metabolome pathway. So we had about 250 metabolites. In addition, the TwinsUK had data on outcomes and some gene expression data. So when we first did the analysis, we got about 69 metabolites, which were significantly associated with blood pressure. And when I say significant, I mean experiment-wide significance. All these are significant. And you can see some of the usual suspects: caffeine, palmitate, which have been previously reported for blood pressure. Hydroxybutyrate has been reported to be associated with blood pressure, lactate, glucose, cholesterol, cortisol. So you have some of these which are known to be associated with blood pressure. We got those signals, and we got a few more, but 69 is too many. We did some data reduction, and we reduced this to about 15 independent signals for blood pressure. They're all quite significant. You can see some culprits. Caffeine does increase your blood pressure. The most significant signal was dihomolinoleate, which was 10 power minus 17, and a few other molecules. The problem here is to try and relate this to causality. I mean, first to try and see whether are these bona fide signals. These are observational data. We got twins. We know blood pressure. There's a big problem with measurement. Lots of measurement error. Lots of confounding in population studies. So are these really true signals? So we thought we'd look at outcomes. We had mortality data. So we looked at mortality. The first thing, caffeine, yes, it does increase blood pressure. It does not kill you. Dihomolinoleate, the best signal for blood pressure, increases blood pressure, but you don't have increased mortality. Whereas hexadecanedioic acid increases concordantly blood pressure and mortality. So, and all the others did not show any relation with mortality. So though the numbers are small, we thought the hexadecanedioic acid probably had the best chance for us to try and take it forward into some functional studies and also to establish causality. So the first thing we did a rat experiment. We increased hexadecanedioic acid by, we got a big bottle of hexa from Sigma and fed the rats. They liked the taste. They ate it. We measured hexa in the blood, and levels increased and blood pressure increased significantly. We then did the other experiment. We increased blood pressure by giving these rats salt, or SHRSP rat salt, to see if just blood pressure increase would increase hexa levels. And we showed no change in hexa levels, but blood pressure increased. So this sort of established a causal link. By increasing hexa levels, increasing circulating hexa levels, we increase blood pressure. We did some telemetry, and we showed if you feed them hexa and increase hexa levels, blood pressure did increase. We took mesenteric arteries. We still didn't know the mechanism. Hexadecanedioic acid is a dicarboxylic acid. It's produced through a minor pathway of lipid oxidation. Most of the lipids get oxidized through beta oxidation. A minor pathway is omega oxidation, which produces the dicarboxylation. And dicarboxylic acids are are water soluble. So we didn't know the mechanism. So we looked at the mesenteric arteries and tested for vascular reactivity, and we showed an increase in vascular reactivity in the hexadecanedioate dioic acid treated rats compared to controls. So that indicated a vascular pathway. Again, luckily, whilst doing the study, we found this paper from Canada wherein they looked at pulmonary artery hypertension. They just did a metabolomic profiling of pulmonary tissue in patients with pulmonary hypertension and controls, and they showed an increase in various metabolites, and one of the significant metabolites was hexadecanedioate acid. So this showed, so using a very agnostic approach, we showed the same metabolite affecting blood pressure in both the systemic circulation and the pulmonary circulation. So in collaboration with Mandy McLean, in Glasgow, who has got a hypoxic model for pulmonary hypertension. So she did the same experiment and showed rats fed with hexadecanedioate acid actually had increased pulmonary remodeling, both in the normoxic model and the hypoxic model. We looked at adipocyte gene expression and found an significantly increased expression of alcohol dehydrogenase 1B. Now that's very important because in the dicarboxylation pathway, fatty acids are first metabolized by CYP4A and then by alcohol dehydrogenase and then aldehyde dehydrogenase. So you need these dehydrogenases to activate the omega hydroxylase pathway, which leads to dicarboxylic acid formation. And why am I saying this? Because that's important in the kidneys, because the first enzyme, the mixed function oxidase, is a cytochrome P450 enzyme, which is present in the kidneys, and it leads to, it metabolizes arachidonic acid to 20-heat. And 20-heat, we know, is a very potent vasoconstrictor. It works in an interesting way. It's a very potent vasoconstrictor in the vessels, whereas in the kidneys, it blocks NKCC2, so actually increases sodium excretion. So there's a balance between vascular function and renal function that can lead to perhaps an effect on blood pressure, and we need to study both. So we are still doing some experiments. We think the adipocyte effect on vascular function could also be related to the perivascular fat producing this dicarboxylic acid and leading to vasoconstriction and high blood pressure. We need to do more experiments to try and do this, but we have potentially identified a fatty acid oxidation pathway or a fatty acid pathway that leads to blood pressure. So in summary, yes, all drugs in general have an effect on renal. I don't think we can discount renal from any pathway for blood pressure regulation. I've shown two examples of untargeted approaches that lead to identification of novel pathways. The GWAS are known to be good at identifying novel pathways for blood pressure. We've seen that for coronary artery disease. I've shown one pathway for blood pressure, and metabolomics is the other one, which seem to be tractable in leading to dissectable pathways. Thank you.
Sandosh, you've indicated that you might try and use the UMOD pathway as a better way to target the use of loop diuretics in the treatment of hypertension. Clearly, we can't afford new expensive drugs in hypertension, but do you think UMOD might be a therapeutic target in chronic kidney disease? Do you think that's possible?
There is a lot of work being done on, because of the original GWAS linking UMOD to chronic kidney disease, there is a lot of work to try and identify how UMOD can affect kidney function. The impact of, furosemide, if you have chronic kidney disease, we do use furosemide for volume control and also for blood pressure. So perhaps in CKD, the effect of UMOD may not be that useful. It's most useful in people with uncontrolled hypertension who have normal kidney function.
Any other questions? If not, thank you very much, Sandosh.